United States Department of Agriculture.
Hi, thank you for the introduction. And thank you, everyone, for attending. All right, great. So let's get started. So today I wanted to talk about organs on a chip technology and particularly the three pillars of organ on a chip technology and where we can innovate. And so again, my name is Vadim Jucaud. I'm an assistant professor at the Terasaki Institute in Los Angeles, where we do biomedical innovation and research. So the first step, or the first thing that I want to mention, is to give you a refresher on what are organs on a chip. So really, those are microfluidic culture devices that recapitulate the complex structures and function of living human organs. So basically, really, it's a microfluidic chip that contains different microchamber and microchannels that allows us to deposit different cells within them and be able to recapitulate living organs. So again, I'm going to go over how we can fabricate those little chips, what are the different materials, what are different fabrication processes, and also going to discuss about what are the types of cells and tissues that we can integrate in those models. And also I'm going to talk also about the control system and monitoring those organs to really be able to do some testing to further healthcare and so on. So let's jump in. So in the first part, I want to discuss really why this technology is really important technology in the future, because it's technology that can recapitulate the complex biological mechanism of organ physiology and disease state in vitro. And this, it can do it a little better than conventional 2D model that don't really recapitulate all the components of the physiology of organs, and also as compared to animal models, because there is always mismatches between species, different animal species, and human, particularly when we want to look at efficacy and human responses. Then also it's important because given the cost effectiveness of this type of technology, it can really accelerate drug discovery and development pipelines. And I think this is really one of the main goals of this technology, is to, again, try to facilitate and reduce the time a drug starts from discovery to translation into the clinic. And another part also is that this technology is amenable for personalized medicine application. And really what that means is that we can use patient-specific cells that we can put in those organs on a chip to be able to look at different drug responses or diseases in a personalized manner that recapitulate all the genetic mutation of a specific individual. So then, of course, the other reason why this technology is really important is because it can be a practical replacement method to improve laboratory animal welfare while maintaining or improving scientific results through accurately representing the human body and generating clinically translatable data. So I think this is very important because definitely in organs on a chip, we can create human organs, which again, when we're going to do some testing in vitro and then translate it into the clinic, we might have a better chance at selecting the right candidate drugs. So now I'm going to discuss a little bit about the different organs that we can model. And of course, we can model virtually any organ in the body, but here are just examples of different tissues that are mostly recapitulated in vitro using this organ on a chip technology. So we have the brain, heart, kidney, bone, blood vessels, gut, liver, lungs, but also other organs such as like the eye, lymph nodes, spleen, skin, or pancreas. And here I want to also highlight that all those organs on a chip models are made to really recapitulate the specific function of an organ. So for example, for a liver, it will recapitulate like metabolism or metabolite secretion. For a heart, usually we try to recapitulate contractility and conduction. With the spleen or lymph node, we try to look at immune responses. With blood vessels, angiogenesis and remodeling. So after all this is really to highlight that organs on a chip, although the ultimate goal is to recapitulate all the functions of a specific organ. So for example, liver may have more than 400 different functions. As of now, is quite complicated to recapitulate all the function, and really organs on a chip are designed as of now to recapitulate a specific function that you want to study or understand better, or a function that when you add a drug, then it can act on that specific function. So for example, for the heart, if you want to regulate the beating of the heart, maybe we can only look at contractility. So organs on a chip technology is made for a specific application. The goal is really to recapitulate the full function of the organ so that we can see things in a more bigger picture. All those organs on the chip also definitely used to study different diseases, so including neurodegenerative diseases, cardiac diseases, liver diseases, inflammatory diseases, infectious diseases, and cancer. And so this is particularly important because those diseases, especially inflammatory or infectious diseases, when happening in human might not really be the same than what would happen in animal models, because definitely there is a huge mismatch, which I can say with the immune system is really where the biggest mismatch is seen, and responses, immune responses in animals really might not be the same at all as what happens in human. And so again, those types of models are really going to be helpful in understanding how human responses are for applying it to the clinic. And so now I want to jump into the place where I call it where to innovate, because this technology was started around 2010. So it's a relatively new field, but it's a very interesting field because it includes many different expertise to really achieve the best organ on the chip to recapitulate organ physiology. So here I want to discuss what I call the three pillars of organ on a chip, which consist of the microfluidic chips, the cells and tissues that are grown inside of the chips, and the control system that allows us to use this chip for some duration and really to be able to monitor and control the models. So in the first part of the talk, I'm going to discuss the design, fabrication, architecture, and different features of microfluidic chips, and also giving you a practical example of some chips that we've developed within my lab at the Terezaki Institute. So then the next part, I'm going to discuss about the cells and tissue, where I'm going to again discuss about the different cell type, function, and then complexity of different tissue that we can integrate into those microfluidic chips, and really also looking at the advantage of stem cells and patient-derived cells. And then in the last part, I'm going to discuss the control system, looking really at automation, microenvironment control, and real-time biomonitoring, because this is also an important and integral part of organs on the chip, because without any good control system, then it's hard to really culture those organs on the chip for a long duration. So again, here is really to highlight that this field is a really multidisciplinary field that includes engineers, biologists, and many other field, and also clinicians, because I think clinicians are very important in understanding what are the types of outcome that they want to see in an in vitro model to really be convinced that this would be a good step towards drug discovery, development, and eventually improving healthcare. So let's jump first into the microfluidic chips. And here I want to really tell you about why microfluidic chip is important in this field. So really those chips are designed to manipulate a small volume of fluid with different channel valves and pumps. And this really is a good approach because definitely we can reduce the amount of samples and liquids and reagents to be used as compared to conventional 2D models. And so those chips with the design, we can create a dynamic microenvironment that closely resemble the fluidic condition of human organs. And also it allows us to have really good control over the experiment, and which allows us to mimic organ level functionality. So here I want to show you different examples of different chip design. So of course, chip design is really limited by the imagination, but at the same time, keeping in mind that we have to recapitulate organ physiology, structure, and function. So here on the left side, I want to show you different simple chip design where you can have single channels where you can perfuse different liquids and you can code the channel with different cells. After here, you have double channels where pretty much you can have two different cell types in two different compartments separated by a porous membrane. And this is the same one here, double channel, but in a sandwich mode. And then we can do it also with multiple channel as well. And so here are different examples. So this one, those two microfluidic chips here are called really a bioreactor. So it's a sort of a generic organs on a chip device in which we can culture in those chambers, which are less than 1 centimeter wide. We can culture different tissue like heart tissue, liver tissue, and so on. And so this really will be more reflective of those single channel type of microfluidic chips. Then here we have another one, those two here, which those are really three or multi-channel chip, which in this case on the right one are used to create different vasculatures or microvasculatures of organ. And this other one here is liver on the chip, actually, where we can put liver organoids and recapitulate the functionality of those cells. And then here we have two other chips that are blood-brain barrier chips, where we can see that we can integrate also sensors and other functionalities. And all this really comes into the design, because before you want to add any other features to this technology, you really have to think about the design of it. So then after the next part, I want to talk about different materials and device fabrication. So there is really many different materials that can be used to create those microfluidic devices, but the most common one is PDMS, glass, and thermoplastic. So those different material really have different pros and cons of using them. And depending on the type of organ that you're trying to mimic and the different function of your organs on the chip, then you would select different types of material. And also the selection of those material will also influence the fabrication process. So the main fabrication process are soft lithography, injection molding. There is other creative methods, but also hot embossing and 3D printing. And really those fabrication method is mostly to create different molds so that we can actually generate those different microfluidic chip with really intricate structures. So the really most promising, or the one that has been used a lot lately, is the 3D printing, because it's really much faster to create molds and to, if we want to do prototyping and iterative process to optimize the chip design. This has really been extremely useful for us to create complex microfluidic system. And so here, this is where I want to describe how we can create those complex fluidic network. And again, with those complex fluidic network, the goal is to have fluid flow control, maintenance of physiological gradient, and establishing a realistic communication between organ compartments when we want to create more complex organs on a chip design that includes different organ modules. And again, these types of design really can help us study organ interaction. And this highlights as well that even though this is design and more of an engineering aspect of the field, of course, it has to always keep in mind how biology works so that we can make a real connections and really understand the physiology of the different organs. So here I want to highlight with those images that on paper, it looks somewhat simple, even though this is kind of a blueprint that is still complicated with a lot of connection. But once we actually translate it into the lab, into a platform, then you can see that things can become a little bit more complex with lots of chips, lots of microfluidic tubing, and lots of connection. And this really is one place where innovation can happen to really facilitate the fabrication and connection of all those organs together, as well as other physical sensors or electrochemical sensors. And so here with that, I want to show you a little example of a model that we made where we, again, thought about the design and wanted to use a new approach to create a blood-brain barrier on a chip. So here in this work, one thing that motivated us to use or to create this new chip was the goal of democratizing this technology. So as of now, this technology is limited to a few labs in the world, and really this is because you need a lot of different equipment, you need some special rooms which are really expensive. And so here, again, when we develop an organ on a chip, One of my goals is definitely to try to disseminate that organ on the chip and being able to have it validated by other groups. And so one way to do that is what we call it to democratize it. So using materials that are relatively cheap. So in this case, we're using a thermoplastic called PMMA, which can be cut using a laser cutter, which is a relatively cheap piece of equipment. And then in this chip, we were able to integrate also sensors or electrodes that we applied using screen printing techniques. And those are carbon electrodes that allows us to measure barrier properties or the resistance of the blood-brain barrier. So in this chip, we culture brain endothelial cells on one side and really try to see, while applying shear stress or flow in the chip, to see how the blood-brain barrier was establishing. What we did, so this just as a schematic, we seed the cells, we apply low flow at the beginning to be able to remove all healthy cells per se. And then after we apply high shear stress or high flow, so which will allow the cell to rearrange and form that strong barrier. And so here, if we can see, we show that the barrier was forming by using immunofluorescence approach. But those techniques are destructive, which again, once you do the fluorescence, you cannot use that chip anymore. So there is a need to actually have some sensor that can non-invasively assess the barrier of the blood or the integrity of the barrier of the blood-brain barrier. And this again, also this highlighting that even though here I'm talking about the chip design, it really encompasses also what type of cells you put in it and also how you're gonna be able to integrate sensor. And so this is what we did. We measured the resistance after 4 and actually went up to 6 days to see how the barrier was improving. And so this is what we see in this graph here, where you can look at the resistance, and as we culture the cells and when we start applying high flow, because again, the endothelial cells that line blood vessels are always exposed to shear stress with the blood circulating within them. So those cells really like that mechanical stimuli, and using the stimuli, we can actually increase the integrity of the barrier. So here we have some staining showing that again, the tight junction were forming in this model, and as well as being a really impermeable model that didn't allow the diffusion of a certain molecule through the model when the cells were present. So with that now, I want to talk more about the cells and tissues, because definitely this is a very important component of the organs on the chip. So there are 3 different types of cells that are mainly used, which are cell line, primary cells, and iPSC. So both have pros and cons. And again, cell line are good because they're highly reproducible, they're easy to use, they're cheap to buy, and they're usually well characterized. But the problem is that they lack physiological relevance. They can have genetic drift, and usually they're from cancerous origin. So definitely to try to study a healthy state of an organ, this might not be the best type of cell to choose. But in that case, you can use primary cells, which have high physiological relevance. You can have diverse phenotypes, and of course, you can generate patient-specific model with them. But the few cons is that those cells really have limited lifespan, that they could be variable depending on how you isolate them and culture them, and also they are difficult to obtain and maintain, because again, to obtain those types of cells, you don't have to get a biopsy or something like that. And then the last one, which are the most promising cell type for this technology, would be iPS cells. And why? Because they're pluripotent. You can generate many different cell types, so therefore many different organs. You can of course make patient-specific model, and they proliferate, not completely unlimited, but they have high proliferation capability. So essentially, there would be a little bit easier to use than the primary cells. But of course, there's another cons that you can have differentiation variability. It could be long and expensive to differentiate the specific cell type in the one that you want. And of course, they could have a genetic abnormality. But then those cells are really used, and this I think is what's very interesting with this field, is we can use tissue engineering approach to be able to recapitulate the physiology of the organs in a better way. So we can use scaffold materials, bioprinting technique, or self-assembly, which can improve the functionality of those tissues. And again, we can have different extracellular matrix, and we can organize cells in a specific way and generate specific function to achieve biomimicry, like contractility, barrier function, like I showed you in the previous model, or metabolic activity. And this here is what you can see in those images. So this is a bioprinted heart, which I don't know if you can see in the video, but this is actually a bleeding. And so those are integrated into an organ-on-a-chip device. Here, same thing with liver cells that are photopatterned in the shape of a lobule per se, and they're encapsulated in a gel which protect them from shear stress, which again allows them to grow well and nicely. Here, it's a picture of vascularization, again, which we are doing within the chip to really have physiologically relevant model that include blood vessels and capillaries. And here is a last figure that shows like some organoids that can be included in those organs on the chip. And this is a liver organoid that still retain CYP function, so metabolism function. And so with that, again, I want to give you another example of how what we generated in the lab, and in this case is a patient-derived vascularized liver on a chip model. So this is a brand new approach where we actually got a discarded human liver that was supposed to be transplanted, but because it was not in good shape, it was not transplanted and discarded. And because the Terezaki Institute has long history in organ transplantation, we actually were able to get some of those organs from which we isolated primary cells and put them into the chip. So of course here we can characterize the cells before putting them in the chip, but the goal here was to really recapitulate liver regeneration. And so we have a vascular model where we create a vasculature, then the hepatocytes start to rearranging within the model, creating buds. Then those buds are starting to become more like lobule, where they become vascularized. And so here is really how it looks in the chip. So you can clearly see that after growing this chip for 7 weeks, which is actually a very long time, and I think this is also one advantage of organs on a chip technology, we can culture tissue and models for up to 7 weeks or even more, I would say. And so here we can clearly see that the top row is the live dead staining. So here you can see that most of the cells are alive, and then you can see throughout time that there is clumps of cells that are forming, like particularly after 7 weeks, this big blue blob, which really corresponds to a liver lobule. So here in the bottom one, we are looking at CD31 staining, which is endothelial cell marker. And we can see at the beginning, they're a little bit everywhere, but they start rearranging in some networks. So then there is more rearrangement in the second week of those networks. And then in the third week, that's where we start seeing in green the hepatocyte or the liver cells arranging into buds. After the fourth week here, we can really see that there is more hepatocyte buds that are formed and attached to the vasculature. And then after 7 weeks, when you see those buds that are white here, is buds that are positive for hepatocyte, but positive for endothelial cells. So here we really have the bud or organoid that is vascularized. So again, this creates really a very functional model where, again, the vasculature is an integral part of the organ, in this case is a liver. And so also what's very interesting with this type of model, so hopefully you can see with this video, but so we can also perfuse immune cells. And so then we can really look at the interaction of those immune cells with the microvasculature, because again, in the body, immune cells usually travel within the blood. And so this is particularly important where with immune response, those types of models will clearly have better recapitulation of what happens in humans as compared to animal models. And so now I want to discuss a little bit more about the control system, which again also is an integral part of this organ on a chip technology, because it allows us for precise and automated control over micro environmental parameter like oxygen, temperature, pressure, pH, or metabolites. And also it allows us to manipulate fluid flow or apply mechanical forces and introduce other clinical stimuli to enable accurate replication of organ function. And here are different examples of sensors that we have generated in the lab to be able to monitor the organs on the chip. So here we have an oxygen sensor that is using a LED light where we can actually see changes in oxygen within the microfluidic chip in a real-time manner. So then underneath there is here a temperature and pH sensor that again is just an electronic chip that we can integrate within the microfluidic chip where we grow the cells. Here in the middle one, this is really to show actuation and how we can easily control and route liquid within different channels. So this is again important to apply different forces or introduce chemical stimuli and so on. And this we can do it in an automated manner using what we call quake valves that can block or open any microchannel. And then here we have another chip with integrated sensor. So here it's also a specific design that allows us to put the probe within the cell culture chamber to be able to monitor dopamine secretion from brain cells. So here again, we are able to really put the probe very, very close to the cells, because definitely dopamine is not going to travel so much far out of the organs of the chip. So this is another approach, again, to be able to monitor responses within the chip. And then after, here's just a schematic of oxygen sensor, and here another microscope that we have integrated, but which is a little bit more complicated, where it allows us to see 3 different color dyes. So again, having this integrated, is really much easier to study all those response without having to place it into a confocal microscope or other heavy equipment. And so here again, I want to give you another example of a model that we've developed in the lab that includes sensors and real-time sensor. And so here we developed a model where we have a liver on a chip model that can be treated with drugs or not. And then here we have a PCTIR sensor that is label-free optical-based sensor that allows us to detect different biomarkers. So in this case, we looked at biomarker of liver injury to be able to assess drug-induced toxicity in the liver. And so here, what we did, again, we created this model that is actually inspired by the architecture of the circulatory system in the liver, where there is 2 inlets, one from the portal the vein and one from the hepatic artery that drains into a central vein. And so in the middle chamber, this is where we culture our hepatic cells that are encapsulated into a collagen gel, which is an extracellular matrix that is highly present in the liver. So what we have shown is that using this type of model, drugs and nutrient and oxygen can diffuse within the model easily, allowing us to culture this model for about 5 days. And here we can see that after 5 days, the cells are growing and again staying alive. Here we look at also again growth and how they are forming sort of like spheroids within the chips. And so those cells remained alive for 5 days. And what we also can see in terms of function, they secrete albumin, which is one marker of function. And when we use the chip with a dynamic system, so with flow, we can see that the cells actually secrete much more albumin. So again, showing that organs on the chip technology are superior than static conventional 2D models to look at functionality of cells. And so here, what we did, we treated those cells with doxorubicin, which is a chemotherapeutic agent that can have liver toxicity. And so what we see again, those are just the standard tests that usually are used to measure toxicity, which is live dead assay using confocal or ELISA. And those are usually done at like endpoint measurement. Whereas here we have this new sensor that allows us to really detect in real time how much biomarkers are secreted by the hepatocytes or the liver cells here. And so here what is nice is that we can confirm that using our sensor, here we don't really see any difference in albumin secretion in response to this drug. So which we also see here with the ELISA, so suggesting that albumin might not be the right biomarker to monitor toxicity. But on the other hand, when we look at GST alpha, which is a liver injury biomarker, we can see that after 24 hours in the ELISA, we can see a difference when it's treated with a 20 micromolar of doxorubicin. But in the case of our real-time sensor, we can detect those changes as early as 3 hours. So again, showcasing that is much faster than conventional models to monitor toxicity with higher physical relevance. And so now the last part that I just want to touch back on some of the challenges that remain. So again, with the microfluidic chip, the main challenges are going to be scalability, reproducibility, and the standardization of fabrication process. For cells and tissue, again, is viability, functionality, and also scalability. And for control system, there is still challenges regarding multi-scale control, looking at like high flow rate or low flow rate and so on, and looking also at dynamic stimuli and also standardizing all those control systems. And so with that, I want to just acknowledge all the team at the Terezaki Institute that contributed to this work and all our collaborators and funding agency that allowed us to develop all those very interesting in vitro models. And with that, I'd like to thank you, and I will take any question.
USDA is an equal opportunity provider, employer, and lender.